thank you all for coming for my PhD thesis defense. I'd like to especially thank James, my advisor, for all the help over the years and getting me through the PhD, and also my committee, Maria, Greg, and Vikram, also for the help in getting me through things. And of course, everybody that has come to see this, both in person and online. And so without further ado, I'm going to talk about my thesis titled Breaking Barriers in Multiscale Agent-Based Models, Effects of Cell Individuality on Viral Infection Treatment, and a Path for Cross-Platform Validation. So as you can imagine from the title, my thesis has two general themes. At the beginning, I focused on building mechanistic agent-based models of diseases, processes, and their treatment. I've explored different hypotheses about treatment, generated new experimental questions. For instance, what can agent-based models bring to more traditional pharmacokinetics models? And then more recently, I've been building infrastructure for cross-platform replication of tissue bio-ABMs and for publishing CompuCell 3D models online. So what are possible difficulties with a universal tissue bio-ABM models specification and a Python port of PhysiCell's phenotypes? And today I'm gonna focus on the second part and two parts of the second part, in specifically, which is the universal model spec and the Python port. So as an outline, generally I'm gonna have two parts for this talk. First, cross-platform replication of cell-based tissue agent-based models, and then templating cell behaviors and how it changes with PhysiCell. So for this part, I've collaborated with James, my advisor, Dr. Macklin, and also Dr. Bruch over at UT Dresden. And we want to make agent-based models more interoperable in between frameworks. So what information do I need to model the function and dysfunction of a tissue? Well, I need to know what cells are, their properties, their volumes, behaviors, how they move, do they chemotax, how does angiogenesis works, and I also need to know how all those aspects break. Does a chemical like arsenic stop VEGF from acting on the cell and therefore stop angiogenesis? Things like that. And so the biology represented by tissue ABMs aims to represent objects, states, dynamics, processes, and interactions of real tissues. And the objects are several. The universe itself that the cells are going to be in, their environment is one. The cells, of course, chemical fields, be it particles or whatever else, molecular networks and other bio-networks that may control cell behavior, like a metabolic network. And then each of those has a state. So a cell can be alive, infected, dead, etc. A field is going to have different concentrations and may diffuse at different rates. The networks are going to have different shapes and rate laws governing them, and so on. Then we also need the dynamics, so how each of those objects evolves with time. What are the rules that determine the next system state from the current state of the system? And as a note, I want to point out that for cells in their environment, there is no inertia. The movement is overdamped because they live in a very viscous environment in regards to their size. So velocity is proportional to force, not acceleration. Then we also have processes and interactions. So the diffusion equation itself, the equations of motion, rate equations, diffusion equation, and so on. And finally, parameters for cell volume, membrane area, the rates that control the rate laws, etc. So an agent-based model has to define how to abstract each of them for them to be useful. And in a simulation in the computer, we don't have anything for free. We have to literally define every single thing that is included. If we don't say something exists, it won't exist. So how do we go from a microscope picture of cells to a simulation of cells moving in this case? Well, for time and space or the universe, you're going to define a volume usually for ABMs, which can be lattice-based or off-lattice. We also need boundary conditions for the simulation. And we need to decide if time is going to be regularly discrete or if we can have varying time steps. Then we have the metadata, which maps the universe, the simulation to real life. We need all the agents, the cells, which can be points, collections of pixels, single pixel. We can have fields as a grid of values or as a mesh and other options. Then each of the objects and the agents, objects, everything else, will have different states with different parameter volumes and rules and instances. Dynamics for all of them, the diffusion equation, equation of motion, ODEs or PDEs for biological networks, and so on. And finally, processes. So rules for mitosis, how that actually happens, for secretion and uptake, and parameters. So today I'm going to use throughout three example frameworks: CompuCell 3D, PhysiCell, and TissueForge. In the first half, I'm going to use CompuCell 3D and PhysiCell mainly. And in the second half, CompuCell 3D and TissueForge. They're all tissue ABMs modeling platforms that work in slightly different ways. So in PhysiCell and CompuCell, they define the behaviors, properties, and everything else in slightly different ways. Both are well-established platforms and both are developed here at ISC, CompuCell 3D by Dr. Glazier and PhysiCell by Dr. Macklin. And PhysiCell's model and specification uses a XML file and a C++ file. CompuCell uses XML and Python. PhysiCell is off-lattice for the cells and on-lattice for the diffusing fields. CompuCell is on-lattice for everything. Units in PhysiCell are defined. Units in CompuCell 3D are not usually defined. So what is the length of a pixel, or what is the length of a time step? Cell type definitions in PhysiCell is a freestanding template, whereas in CompuCell it's not unified into a single description. CompuCell has a detailed cell shape, and in PhysiCell, cells are points and a sphere of influence. And dynamics in PhysiCell are force-based. In CompuCell, it's energy-based. Both use SBML for network models. So changes in biology and real life and everything else, why won't you play, can be continuous, which you should see a cell moving here on the left, or discrete, which would be things like cell division or cell death. And that's also true for real life. Birds flying is a continuous dynamics. Then being killed is a event. Oh, I see. Anyway, so how are dynamics in PhysiCell and CompuCell 3D? As I mentioned, in PhysiCell, cells are points with a sphere of influence around them, which gives them a volume. And the cell position update depends on the velocity. And the velocity is updated from the forces acting on the cells. And then stochasticity on the simulation is added into the force. So for instance, this cell red is that point in the center, and this red circle, it's its sphere of influence. And it's interacting with its neighbors, and it feels an attractive force and a repulsive force to each of them. In CompuCell 3D, cell position update is local to the boundary of objects and is naturally stochastic. And the velocity update, the velocity is given by the position update instead of the other way around. So how can we use ABMs within the scientific method? Well, in science, we start by making an observation of a natural phenomena or problem. Then we question how that should work. We create a tentative explanation that we can test. We make a prediction, and then we go to make an experiment. In our case, our experiment is our simulation. So it's an in silico experiment. We create an agent-based model that reproduces the observation and can test the hypothesis. Then we compare the in silico results with wet lab experiments if they exist. Hopefully they do. Then we go to make the analysis and conclusions, and we can suggest new wet lab experiments, share our results and the model with the community, and hopefully reproduce the model using another method. So do ABMs follow the scientific method? In general, no. Science and computational models should be fair, findable, accessible, interoperable, and reusable. Tissue ABMs usually do the first two. They are findable and accessible, but they lack the later two. They are not interoperable and they are not reusable. I can't just pick a PhysiCell model and put it in CompuCell and run. To do that, I need to reinterpret the model and rebuild the model pretty much from zero. So this slows down research, of course, inhibits collaboration, and causes a crisis of reproducibility. How can we change that? One way to do it would be to have a universal interoperable tissue ABM specification standard. This would fit many frameworks at once. So we develop this specification once, and each framework would read it in and run the simulation from there. It would be responsible for describing the metadata, the universe, all the objects, initial conditions, and everything else. And it should also be expensive so that we can add in more behaviors and methods to it that then can be adopted. And it should be object privileged, meaning that it should privilege what a cell is instead of how a cell moves, and things like that. Because a cell is going to be a cell, is going to be a cell everywhere. How to make it move, as I just said, changes. So what steps can I take to achieve the universal spec? First, can I develop this myself? Well, it's not a single person project. It's going to affect many ABMs groups, so they should all have a voice in how things will work and go. There are many unknowns with building something this broad, and it's simply a pretty huge task. But is there any intermediary step that I can take? In that case, yes. I can make a translator to go from PhysiCell to CompuCell. And with that, I can uncover some of the challenges that the universal spec will face later. And I can already try to find some possible solutions for it. So what does the translator do? It needs to read the original specification, interpret how information is organized and defined, find solutions for missing concepts, which is the big one, and let the user know if any issues were found and what is not translated. Then write the resulting specification file or files and keep up to date with both target platforms, because as they change, everything about the translator is going to have to change. So a little bit about the model structure in PhysiCell and CompuCell. As I mentioned, PhysiCell uses XML, which we have here, and C++. So here we have the definition of the worker cell in this model. It has a migration speed, a migration bias, and chemotaxis information here, and some other things that I've cut off. And here is the C++ that has more specific behaviors for it, because this is a complicated simulation. So it needs C++ on top of everything else. So event rules in PhysiCell are or were defined in C++, but I believe Paul and his group are working on making that part of the XML too. In CompuCell, it's again XML, but now we have Python. And here I have the cell type definition for all my cell types. We see that it's way simpler and we just have the names of each cells. And then chemotaxis is defined here. We define which fields can have chemotaxis. And finally, in Python, we actually activate and deactivate chemotaxis and switch chemotaxis type. So in a bit more detail, my translator does things in a few steps. First, it parses PhysiCell's XML and re-implements the information in a manner that can be used in CompuCell. And it automatically translates the cell types and their constraints, the cell behavior embedded models, which I'll get to in the second half of the talk, the fields information, chemotaxis information, secretion uptake, and boundary conditions. Things that it doesn't translate yet are the initial spatial cell configuration and how to transition from one set of cell behaviors to the next. And of course, anything that was hand counted in C++ won't be translated. So the actual steps are to first read the XML file. Then I use a Python package, XML to dict, transform the XML into a Python dictionary. I just found that easier to work with. Parse the dictionary to extract and organize the data, and then perform string operations on the extracted data to generate the XML. And finally, write the resulting file. So here I have two functions that write that cell type plugin for CompuCell that I showed a few slides earlier, where here I have the make cell type plugin. I have a header, and then it passes the dictionary to this other function, make cell type tags, which in this part loops over all of the cell types and extracts their names and creates the XML tag. And then all the XML tags string is returned, a flag for having a wall around the simulation, and all the cell type names as a list instead of being part of a huge string. So a few comparisons of translated simulations. So here we have a cell cycle model in PhysiCell and CompuCell. Cells are going through the G1, S, G2, and M phases. And same here in CompuCell. The only things that I changed for the CompuCell simulation was to make the cell a bit bigger to make it easier to see, and the color map for all of the phases. Another example I have is the biorobots simulation. Again, here in PhysiCell, here in CompuCell straight out of the translator, and here with the extra steps I took. So the worker cells, I actually showed the C++ for them earlier. They chemotax in two different gradients, one that is secreted by the director cells and one by cargo cells. So cargo cells are blue, worker cells are red, and director cells are green and a bit hard to see, but there's one just left of my pointer there. Worker cells chemotax trying to find cargo. When they find cargo, they attach to it, and then they start to chemotax on the director signal. And when they find the director, they drop the cargo. And the same is happening here. The only real difference is that director cells are in yellow. And I also, to make the cargo cells not move so much, I switch their types to director cell when they are dropped, but they don't secrete the chemoattractant. So how can you use this software? You download it from the GitHub page, which I have a link to it later, and then you run this convert_by script using Python. And it has 3 switches, optional switches for having a minimum cell volume, which we'll get to why later, a maximum simulation volume, and the desired output directory. And the only required input is the path to the PhysiCell XML you're trying to translate. So when actually using this, it spits out a lot of debugging data and lets the user know about warnings that may happen. This one is about the cell volume being too small, and this about the simulation domain being too big. In the end, it tells you when it's generating all of the files and where it wrote the files. So a bit more on how the code for both look. We have the XML that defines time and space for PhysiCell here and CompuCell here. So in PhysiCell, again, for cells, it's off lattice, but for the fields, it's on lattice. So it has a discretization constant here for the fields, but the overall volume is defined here. And here we have the units for time and the units for space, and also the time steps and the maximum simulation runtime. And then here in CompuCell, we have the dimensions of the volume in pixels and the number of steps it needs to run for. And I want to point out these other XML strings, which were all added in by me. These comments are to guide the user if there are some concepts that are in CompuCell but not in PhysiCell. And these ones are to add in the information for what the units should be in CompuCell. Since PhysiCell has them, I thought it was nice to actually make them available in CompuCell. So this one defines how many microns a pixel is, and this how many minutes a time step is. And a time step in CompuCell is called a MCS or Monte Carlo step for historical reasons. And of course, I also make these conversion units available inside the Python. So each of the Python parts of CompuCell has it available. So then we also have the cell volume constraints, where here we have the volume for the director cell from before, which is about 2,500 microns cubed. And in CompuCell, I keep that information associated with the cell in its cell dictionary, which is used by cells in CompuCell to have any sort of data associated with them. And then the volume in pixels is actually set to the cell, and so is the strength of the volume. And we can see in the PhysiCell example that we also have a nuclear volume and a fluid fraction, which are two concepts that we don't have in CompuCell. And when we have something in the target platform, but then not in the original, it's easier to deal with than when we have something in the original and not in the target, right? So that brings us to translation challenges, right? The translator work, what were the problems and solutions I found? So there are actually two types of challenges: translation issues and optimization issues. The translation issues are things like defining pixels and time steps, how to interpret concept definitions and concept disparity. So how many microns should a pixel be? How many minutes should a time step be? And things of that nature. And then dealing with missing concepts. So all the behavior models that PhysiCell has and CompuCell doesn't. And things like all the different volumes that are in PhysiCell but not in CompuCell. And then we have optimization issues, which is how to select the pixel size, how to select the time step length. And these mostly arose from choices I made earlier in the translation. If I had made different choices, my life would have been a bit easier. And the main points of difficulty that I faced were representing the cell behavior sequence models and the optimization issues. So how did I choose the pixel size? I need to relate a pixel in CompuCell to the off-lattice cell nature of PhysiCell. So I decided to actually use the diffusion because that has a lattice, so it would be easier. And I use it in PhysiCell's lattice constants, delta x PSC, to discretize space. And that will define the conversion and how many microns a pixel represents. And so converting spatial parameters, we'll use that in. So the volume of a cell in CompuCell is the volume in PhysiCell in microns divided by this discretization constant, which has units of microns per voxel, to the dimension of the simulation, so either 2 or 3. But that choice caused my first optimization challenge, which it makes my cells in CompuCell be less than 1 pixel, and that's just impossible to represent in CompuCell. To solve that, I stretch my simulation in all directions so that I have a finer granularity of the simulated domain, and so my cells actually have more than 1 pixel. So instead of using delta PSC, I define a delta CC3D, which is a constant less than 1 times the original delta. But that actually brings me to another issue, which is expanding the domain side. I may have an overflowing number of pixels, so more than 2 to the 64 pixels. That happened quite a bit. So I decided to, and even if I don't reach there, if I have the cells and fields, I end up using a lot of memory, so over 10 gigs of RAM, actually more than 20. And so in that case, I truncate the simulation, cut off parts of it, diminishing the overall domain size, which does mean that I may miss some important spatial information from the original model. For instance, if you have a tumor near some artery and you're trying to simulate angiogenesis, maybe I cut off your tumor or your artery. So next was translating time. So the time step in CompuCell is, in general, 1 Monte Carlo step. So I'm defining the time step in CompuCell to be 1. And in PhysiCell, the time step is X amount of minutes, usually. So I want the total amount of time both simulation run for to be the same. So I need to find out how many time steps PhysiCell runs for and use that as the number of time steps for a CompuCell. So the total runtime of CompuCell divided by its time step, which is 1, should be the total amount of time in minutes in PhysiCell divided by its time step. And as the time step in CompuCell is always equal to 1 in my head, I found it more natural to define how many time steps is a minute instead of how many minutes is a time step. And that, in the end, is just 1 over the time step in PhysiCell. And then I use this row here to do the conversion of any parameters that have time with them. Which brings us to the next challenge, optimization challenge, which was the diffusion. So the diffusion parameters both have time in them. The diffusion constant is length squared over time, and the decay is 1 over time. So to convert the original PhysiCell one to CompuCell, I divide the original by the row, which is the number of steps that a minute has, and the space constant, rescale the space constant squared, and similar idea for the decay constant. But if I do this, each of my CompuCell time steps takes too long to run. And that's because PhysiCell does diffusion in a very coarse lattice. So it's end up being faster than CompuCell, which uses the same fine lattice for the cells and for diffusion. We've actually been talking about maybe doing some coarse screening and diffusion for CompuCell too. So to not wait forever for each time step, I redefine row so that to increase the number of steps that are a minute and then decrease my diffusion parameters. And then we now finally have the biggest one, which was the concept mismatch. CompuCell usually thinks from the point of view of the universe, so from processes, so from the movement of cells and energies acting on cells. And so the cell definition is spread out among different parts of the model spec. And PhysiCell is more object, the objects are more prioritized. And so in PhysiCell, a cell of a given type has all of its behaviors that are associated with it in a single template. And so the second part of my talk is me dealing with that. But on the other hand, and PhysiCell cells don't have a real volume of surface, so they don't have cell-cell contact. They use long-range forces to approximate that. So the universal spec would have to define things in a way that it can be read by anybody and should be designed to be expensive to include more behaviors as it evolves. So in conclusion, for my translation, after I've successfully built a translator that generates a CompuCell model from a PhysiCell model, and I found solutions for both the translation and optimization issues that I found. Now replication of PhysiCell models in CompuCell is easy, or at least easier. And the translator serves as a proof of feasibility for the universal spec. And my work in this is going to be used as a springboard for Dr. Macklin and Dr. Glazier's new NSF post grant on building a standardized ecosystem. And here I have the link to the translator. So it's GitHub, my name, -PCXML2 CC3D. So PhysiCell XML to CompuCell 3D. The next steps for the translator is to improve the call commands. I want to have a few more switches for different things. Improve the default initial cell configuration actually translate the initial cell configuration from PhysiCell. And I'm working on an article describing the translator and how it works. And I've came up very recently with a mock of how the universal spec could look using YAML, which is a machine readable language that is more modern than XML and easier for a human to read too. So for the second part, it's templating cell behaviors and how it changes, PhenoCellPy, which I have is a Python package that I created, and I have a preprint out for it. And it reimagines the behavior models from PhysiCell. And for this, I've collaborated with Dr. Sego from the University of Florida, Dr. Slupka, which is also at my lab, and Glazier. And the point of this is that in biology, cells and humans have different behaviors, types of behavior. They are of different types. They have different behaviors and sequences of behaviors. So a behavioral sequence for a human would be the Sphinx's riddle, for instance. And for cells, it can be things like the cell cycle or the stages of viral infection, stem cell differentiation, and so on. So in each case, we have behavior changing in a patterned way. And what causes the behavior to change will be highly dependent on the biology being modeled. Has the cell finished its pre-division growth and internal reorganization? Has it been infected by a virus? Has it found a food source? So if we want to abstract all of this, we need to somehow pattern it all. And this pattern is going to be made of two parts: the template of behaviors and interactions, and also placeholder states for all of them. And then a set of rules that define when the transition between states happens. And we actually do this all the time when we build biomodels without even realizing. And so it's actually kind of wasted effort by the community because we are reinventing the wheel a lot. So if there was some way to standardize the pattern abstraction, we could share the pattern and improve replicability and improve the community. So PhysiCell has an elegant way to do the templating. The whole pattern in PhysiCell is called a phenotype, and each state is called a phase. So, and I'm adopting those as my nomenclature. So the whole cell cycle would be a phenotype, and G would be a phase. And since this is so elegant, it shouldn't be locked into a single platform, and I want to make it available to more platforms. That's why I came up with PhenoCellPy, which reimplements all the phenotypes model from PhysiCell and integrates with other agent-based platforms. PhenoCellPy models are embedded models, so they are not going to replace the agent of the platforms or replace anything in the platform. They're going to be inside the agent on a platform. So we have the main model. Inside the main model object, we have the agent object. Inside the agent object, we have the phenotype, and then some more objects inside the phenotype. I wanted to make it easy to generate new phenotype and phases and to have good default parameters for everything. I also wanted to make things highly customizable and have minimal software requirements and make it easy to install. So what are my initial target platforms? Since I'm in Python and since PhysiCell already has phenotypes, it doesn't make sense to have PhysiCell as a target. So my targets are CompuCell, again, and TissueForge. TissueForge is a center-based Python model developed by Dr. Siegel over at Florida. And it also does tissue ABMs. But in the end, I want to make this available to any Python platform. So I'm using these two as testbeds and to develop APIs for them to make things easier for the user, and then eventually making a generic API that can be easily customizable to new platforms. So the phenotype class, it has several attributes and functions, like the name for the phenotype, the time step length, and so on, and also a time step function and some other functionalities. I want to point out a few of them. The attribute I want to talk about is the senescent phase, which is not the greatest of names. I'm planning on changing that, but it's a special phase on the phenotype that has no exit and freezes parameters, right? You can think of phases that can have either one of those. This has both. For instance, when a cell dies, it can't become alive again, so it has no exit, but it's still its parameter and dynamics are not going to be frozen. It's going to dissolve into the environment, for instance. In another case, can't think of an example right now of the dynamics freezing, but maybe a red blood cell has its dynamics frozen, but it may exit somewhere. Then we also have to have dynamics associated with all of it. The dynamics is mainly going to live in the phase, but as a convenience, if there is some behavior that spans the whole phenotype, the user can define that in the user phenotype time step function for the phenotype. Speaking of the time step, here it is. It does quite a bit. So I'll take it in parts. But what we can see, again, is the hierarchy of things with the main model, the agent, and the phenotype. So the phenotype time step is actually called by the agent itself. And eventually the agent is gonna get back some flags for what happened this time step with the phenotype and what should happen with the agent later. And right now, what happens to the agent based on these flags is done by the universe, and you need to have some watcher on the universe and some code to do things. The first thing the time step actually does is called the user phenotype time step. So the general dynamics for the whole phenotype. Then it calls this phase that it is in time step, which returns flags for going to the next phase or exiting the phenotype, which would be going to that senescent phase. And then if phase switch is signaled, the time step calls the go to the next phase method, which checks for division at phase exit and for removal from the simulated domain at phase exit, which can be interpreted in many different ways, be it migration out of the simulated domain or death or something else. And also calls the next phase entry function. So a cell when entering G is going to grow its volume. When it goes to M, it's going to divide. If senescence was signaled, it calls the go to senescence phase methods, and that method freezes all the dynamics and parameters. Then finally, it returns to the agent the flags for changing phases, cell removal, and cell division. And then the user is currently responsible for calling their platform's methods for what to do then. For the phase class, we actually have a lot more attributes. Some are similar, like name, time step. But there are more attributes here because things like volume dynamics are all a property of the phase. What volume the cell should have, or its target volume, it's going to depend on the phase. It's going to be higher in a G phase than it was in S phase. And also how fast the volume can change, or biomass change rate, is also a property of the phase. And I want to point out that there are some entry and exit functions that happen on phase entry and on phase exit, or just before say phase exit, as conveniences for different behaviors. And also for triggering the volume expansion or triggering mitosis and so on. Then the phase time step. This one is a bit simpler than the phenotype. And it will first call the cell volume update, which is also part of this phenotype. We have a controller for the cell volume. Then it calls the user-defined dynamics for this phase, checks for phenotype arrests or senescence, and returns early. And then checks for phase transition. And during the check for phase transition, it also can check which phase to go to, because you can imagine that one phase can have different exit points. And returns the flags that we talked about earlier to the phenotype time step. So, as I mentioned a while ago, how phase change happens is highly dependent on the biology being modeled. But I want to give some good defaults to users. And the two generic methods that I have are based on the time spent in a phase, the phase expected duration, and the time step. And even though the cell doesn't know how much time it has spent in a phase, it has some marker that it can use for that, even be it its volume, some enzyme level, or something else. And that can be abstracted as a timer. And besides, it's also a useful datum for the modeler to know how much time a cell has been in a phase. And so these two methods are stochastic and deterministic. In the stochastic case, we have a fixed probability to go from phase A to phase B. And in the deterministic case, we have a fixed time duration for phase A. And I want to note that for the stochastic case, I can't approximate this Poisson event as the usual time step over expected duration, because I don't know what time step the user is going to use or what expected duration they're going to use. There is a included cell volume within the phenotype, and that was in the original PhysiCell phenotype, so I wanted to be thorough and have that here too. And I believe that was built into PhysiCell because the phenotypes began as to deal with cell cycle. And this class segments the cell volume into 5 parts. The cytoplasm, both a fluid and a solid part, the nucleus, a fluid and a solid part, and a calcified fraction for the cell. And each of the volumes does a exponential relaxation to a target volume, which is a property of the phase, the target volume is, and the biomass change rate is also a property of the phase. I have some pre-built phenotypes, the same that PhysiCell has, Ki67 basic and advanced, flow cytometry basic and advanced, and 2 death models, apoptosis and necrosis. So Ki67 basic is a 2-cycle model, which uses Ki67 as a protein marker for cell proliferation. So if the cell doesn't have that marker, it's in a quiescent state. If it does have it, it's proliferating. The advanced one adds a post-mitosis phase for degradation of Ki67. Flow cytometry is the classic G1 as G2 and M together and back, and the advanced separates G2 and M. Then apoptosis standard is a single cell phase for cell death, where the cell dissolves into the environment. And necrosis is a two-phase that phenotype that represents first the hydropic swelling of the cell as it shuts off and eventually it bursting. So I have two examples of PhynoCell running within the target platforms. This is TissueForge, and I'm running the Ki67 basic. And I have the volume of the cells pegged to the volume model from PhynoCellPy. And we actually can see them grow, although it's a bit hard to see in this movie. And for this, what I had to hand code was just the volume matching and calling the division on the cells when they divide. The second example I want to show is the necrosis phenotype in CompuCell, where I selected the green cells to undergo necrosis at the start of the simulation. Their volume is again controlled by PhynoCellPy, and we see that they swell up at first, and then they burst, lose a lot of their volume, and slowly dissolve again. So hand-coded the volume matching and switching cell type. So PhynoCellPy's next steps, I divided into a few groups. I consider it PhynoCellPy to be in alpha. So for the beta release, I want conda distribution. I want to finalize the APIs for CompuCell and TissueForge, finalize calibration, and be able to randomize the initial phenotype phase. For 1.0, I want to improve code architecture, add parameter heterogeneity, have the generic API for other platforms, and add APIs for volume, pegging, and so on. I also want to add concepts of parallel phenotypes, so you can have a cell that is cycling and is infected, for instance, and also phenotype switching, so going from being alive to dead. And also add some more pre-built ones. And other features that I want to add is to add a membrane model for both division and for the necrosis. I think it would be fun to have membrane tension control the switch there. And add methods for time to event modeling approach, which would be instead of having a fixed probability to go from one phase to the next, I have a varying probability. And I also want to add just-in-time compilation to all of this to make it even faster. So in conclusion, I've reinterpreted, reimagined, and reimplemented PhysiCell's phenotypes models. I've made them accessible to more modeling platforms. I've built a roadmap for its near future development. And I hope it will be part of the platform interoperability efforts being spearheaded by Glazer and Macklin, because we already have SBML to abstract all the bio networks. Hopefully this can abstract the cell behavior patterns. And PhynoCellPy is accessible, intuitive, and enables modelers to add details to their model without much overhead. So that concludes the talk, but as general conclusions for my time here at IU, I'd say I did well. I have 4 peer-reviewed papers out and 3 preprints. I've built collaborations all around the globe with people, a lot of labs here in the US, people in Germany and Australia. My research has addressed important questions about mechanistic agent-based models and how they can be used and developed to study infection in tissues, which is something that if I had 3 hours, I could have talked about. And my translator successfully converts a PhysiCell model into CompuCell, and PhynoCellPy makes PhysiCell's phenotypes available to more platforms. Now I'll just give you my references. If you can read 1,000 words per minute, maybe you can see something, but reference 1, 2, 3, and 4. And for my next steps, I'm starting a postdoc with Dr. Amber Smith over at UTHSC sometime in August. And I'm gonna model pneumococcal pneumonia co-infection modeling, and many more fun stuff. I want to continue PhenoCell Pi's development on my off time. It's my baby. And I want to help with the Universal ABM Spec consortium. So I like to thank you all online and in person for coming. I like to especially thank James for all the help during the years, the suggestions that were always good, the lending a year when things were tough, and my committee, again, Maria, Greg, and Vikram for the help to getting me through this phase. All my amazing collaborators over the years and coworkers. If I missed you, I'm very sorry, and I'm gonna say that I couldn't find your picture and it's your fault. I also want to thank my funding agencies for keeping the lights on, both at home and in the lab, NIH and NSF. And last but not least, my family, friends, and everybody else that made me be able to achieve this. So thank you.
Are there any questions from people who are on the Zoom session or in the room which are not on the committee? Feel free to unmute and just ask the question if you're online. Anyone here? No. Okay, then I need to ask the people who are not committee members to log out or to leave the room for a second. And the committee then will have its chance to ask questions, okay? Thanks everybody for coming.